Okay, so we have our prophase group. So who was going to be our speaker for our prophase group? And you're able to share your screen.
That's me.
Okay, go ahead.
Okay, so this is our first question. Then it's what's the number of the cycle that corresponds to our stage, which is prophase. So prophase is the first step of mitosis. So put 1. And then with the main function of our stage, put during prophase, the complex of DNA and proteins contained in the nucleus, known as chromatin, condenses.
Okay.
And then our second was outline how chromatin and chromosomes that consist of identical sister chromatids. And then I would put the cellular DNA is replicated during interphase, resulting in the formation of 2 copies of each chromosome prior to the beginning of mitosis. And then as the cell enters mitosis, chromatin condensation leads to the formation of metaphase chromosomes consisting of 2 identical sister chromatids. Then our third question is, explain why the process of mitosis results in cells that have the same combination of homologous chromosomes. Because the genes contained in the duplicate chromosomes are transferred to each other, successive cellular generation, and all mitoprogeny are genetically similar.
Okay, very good. Thank you so much. All right, you did great. Let's see here. Try to turn up my volume so I can hear y'all. Okay, good job. Good job for prophase. Okay, all right. So okay, with prophase, we talked about how so those chromosomes are going to start to condense, and they, you start to be able to see them. So what else happens as far as the nucleus goes? What happens to the outside of it? It dissolves. Yeah, so your nuclear membrane is going to dissolve. Okay, and then additionally, we have another organelle that is very important when it comes to mitosis, and it's going to start to produce spindle fibers. Okay, so we have our centrioles that are moving to the side. They're going to start to produce spindle fibers because those are going to attach to the centrosome, what's it called, to the centromere of that chromosome. Okay. Very good. All right, so let's see here. Nick, you're the only one that has pro-metaphase. I'm sorry about that. I don't know if you're able to share so that way you can show us what you figured out.
Yeah, I can share.
Okay, and you only need to go over number 1 because I think questions 2 and 3 are the same for everybody.
Okay. Okay. So my phase will be here in stage 2, and specifically in it, as the chromosomes condense, kinetochores appear in the centromeres, and basically these mitotic spindle microtubules attach to these kinetochores, and these centrosomes move towards the opposite poles.
Okay, very good. Yeah, so I like to think of kinetochores as like little handles in the middle of the chromosomes. It's kind of how they draw them. And so that's what those spindles attach to. They attach to those little handles, so that way it can pull the chromosome apart. Okay, very good. All right, who had the third phase?
I did.
Okay, are you gonna present?
I can present. Yeah, no problem. Okay. So let me just share.
Okay. You only need to do question 1.
Okay. So we had metaphase, which is the third stage. The function of metaphase is the microtubulars pull the sister chromatids into an aligned formation, as you can see right here, which gets the cells or the cell ready to split into 2. And did you say only do the first question, or can I continue?
Only do the first question because the second and third are the same for everybody.
Okay.
Okay, very good. All right, and so just as a side, so when you're talking about metaphase, okay, so you have your chromosomes lined up in the middle. What's a name for that where it's lined up in that equator? I don't know if it's a vocab. I think it's one of y'all's vocab words. So when the chromosomes are lined up in the middle during metaphase on the equator, what is that called? Yeah, so a metaphase plate. Yeah, good. Yeah, so a metaphase plate. Okay, so it's a metaphase plate where those chromosomes are lined up in the middle. At that point, are they attached to the spindle fibers? I can't remember if Mia said that or not. Are they attached to spindle fibers?
Yes.
Yes, they are. Okay, very good. All right, so who was phase number 4? Yes. Uh-huh. Let's see here. I think if you, because you can screen share, right? You can, yeah, go ahead and plug them in just so that way we can hear you. All right, we're gonna try this. We'll see how it goes. Here, let me turn this down because it'll be it'll like echo. All right, yeah, you're still muted.
Yeah.
There we go.
Okay. Hopefully everyone can hear me. We had Anaphase, and that is the 4th phase. And in Anaphase, the cell is elongated, chromosomes are broken apart, or the sister chromatids are broken apart. Oh yeah, and then they're moved to opposite poles. And yeah, so that's what it looks like.
Excellent. Okay, so there's one more thing. So if you want to go up to that picture really quick.
Oops, I just stopped. Oh no, I didn't.
No, you're still sharing. Okay. All right. So if we're looking at those spindles, okay, so some of those spindles are not actually attached to any chromosomes. Okay, so what is the purpose of the ones that are not attached to chromosomes?
Do they kind of like guide them in a way?
Yeah, so they're going to kind of guide them. What else is going on with the cell just looking at it?
Oh,
Elongation. Yeah, thank you, Ayana. Yeah, so elongation. So they're getting, it's causing that cell to get longer. And so that way, you know, it has sufficient space to divide without actually cutting into those chromosomes. Okay, very good. Thank you. Thank you too. Okay, so I think Danae got booted off and hasn't been able to get back on. Okay, so let me see here. Let me see if I can pull up, if it'll let me pull up Telephase. Nope, I got for a Metaphase. Okay, let me just pull it. Let's see here. I just want the picture. Okay, here we go. Prophase, okay. Okay, goodness. All right, so Telephase. Okay, so hopefully all of y'all can see that. Okay, so Telephase we know is step 4. So based off of this picture, what's going on with Telephase?
The sister cells are beginning to split apart.
Okay, the sister cells are beginning to split apart. Very good. Okay, so do we have chromosomes at this point?
No.
No. Okay, so our chromosomes are decondensing. Okay, so they're decondensing, going back to chromatin. Okay, and what's around my chromatin? Nuclear envelope reforms. Perfect. Okay, and so my nuclear envelope is going to reform around those chromosomes. Do I have my spindles anymore?
No.
No. Okay, so my mitotic spindles are starting to break down. Okay. All right, good job. Okay, so those are my main characteristics for Telephase. So my last phase is cytokinesis. Okay, so one of my speakers wants to go over cytokinesis with us. So I had Dylan, Alexis, and Anastasia.
I can go ahead and do it.
All right, thank you, Dylan.
Okay. So it's the 6th stage, and it's to separate the cells in half and ensure that one cell ends up with a nucleus in each daughter cell.
All right, and so we're looking at a picture. So the picture that we're looking at here, I'll share mine. Okay, the picture that we're looking at, is this an animal cell or a plant cell? It's an animal cell. Okay, so what is it called when cytokinesis is occurring? What is the structure where it's pinching off? Yeah, a cleavage furrow. Okay, yeah, it's a cleavage furrow. So what about for a plant cell? What would it have? A cell plate. Very good. Yeah, so plant cell, you would have like that building of that cell wall in between. Okay, excellent. Great job. Yeah, so y'all did very good. Okay, and I think, let's see here, for y'all's questions, y'all were doing really well. So outline how chromatin, because a chromosome that consists of identical sister chromatids. Okay, so we know that they go through DNA replication, and there's a lot of things that regulate that just to make sure that the proper, DNA is being replicated. So like, for example, your DNA is twisted around your histones. The histones actually have to unwind to go through the process of replication. And then they're going to wind up again just to make sure that they're, tightly coiled and out of the way. Let's see here. Explain why the process of mitosis results in cells that have the same combination of homologous chromosomes. Okay, so same combination. I don't quite understand. Let me see here. This answer doesn't make much sense. Okay, so essentially, so when you have your homologous chromosome, what is each half of that chromosome called? Chromatids. Okay, so your chromatids are going to be what ends up separating during mitosis. So that way you have an even number of chromatids, which was part of your replicated chromosome, in each part of your new nuclei. Okay, very good. All right, so were there any questions with this? You did great on this. I feel like this unit's a lot easier than the last unit. Okay. All right, well, I'm going to go ahead and let y'all go 10 minutes early. Tomorrow we're doing a learning catalytics. Yay. Okay, so I will see y'all then.